our last speaker, Richard Lange from University of Rochester, that will have a surprise Halloween costume in a bit. And he will talk about a study on the macaque visual cortex where they use both electrophysiology and histology. I really look forward to that. Richard?
Yes. Hello, everyone. I wanted to add an acronym so I could join in with the rest of the session, but moving on. So just to start, I want to acknowledge collaborators and funding upfront because this was very much a group effort. So there's everyone. So our goal with this project is to further understand the relationship between a neural population sensitivity to a stimulus, neurons' choice probability, and correlated variability between neurons, as well as how the latter two depend on feedback to sensory cortex. Linear decoding models predict that choice probability depends both on neuron sensitivity to the stimulus as well as on the correlated variability. And indeed, numerous previous empirical studies have reported an association between choice probability and sensitivity. Other studies have found task-dependent structure in noise correlations, suggesting that feedback must be involved. And these task-dependent changes in noise correlations are not easily explained by any feed-forward decoding framework. In separate theoretical work, we were able to derive the optimal form of feedback to sensory areas by assuming they are involved in the hierarchical inference process, where feedback communicates prior expectations. One goal for this theory was to give a unifying account of why F-prime structure appears in both choice probabilities and in noise correlations. So to briefly summarize the entire work, we set out to test this theory and to extend previous experimental results on feedback-induced F-prime structure in choice probability and noise correlations, but we ended up failing to replicate the main effects, which raises questions about methodological differences as well as idiosyncrasies between brain areas, neuron types, or even monkeys. So over the next few minutes, I'll tell you about the experiment design and analysis methods we used to arrive at this failed replication. So we recorded from macaque V1 Utah arrays while monkeys learned two coarse orientation discrimination tasks. The first is a cardinal task in which they discriminated from horizontal from vertical patterns. And the second was an oblique task in which they discriminated diagonal patterns. An important feature of the stimulus is that it's identical at zero signal for both tasks, allowing us to isolate the effect of changing the task cue while holding the stimulus constant. We repeated the same training protocol for two monkeys, monkey U and monkey A. Both monkeys began learning the cardinal task, then switched to learning the oblique task. They then learned to randomly switch between the two tasks based on a cue at the start of each trial. Monkey A underwent the entire protocol twice, once in 2017 and once in 2019. We analyzed data from between 5 and 20 sessions at the end of each experimental epoch, once the monkey's behavior had stabilized. Both monkeys did learn to perform the discrimination tasks well. Here I'm showing psychometric curves for monkey U during interleaved sessions. Monkey A similarly learned the task, but with less overall sensitivity and more biases, likely due to the more peripheral stimulus. In separate behavioral analyses, we found that monkey A did not significantly switch his strategy based on the cue, but only in 2017. So in the interest of time, I'll move on to neural data. Neural data were collected by 96-channel Utah arrays and planted in V1. And unlike previous studies, we decorrelated voltages across the different channels by ZCA whitening and found that this dramatically reduced artifacts and correlation estimates. For further analysis, we counted all spikes per channel in a window after stimulus onset. We did not spike sort because all previous studies on this topic have reported no difference between single and multi-units. We then fit a non-parametric statistical model to spike counts across trials, which we use both to subtract off slow background fluctuations and for automatic outlier removal. In the end, this only removed a very small fraction of the data. In the end, our neural data appeared pretty much as expected in terms of orientation tuning, PSTHs, tuning to the signal in the task. Here I'm just showing a few example neurons. And noise correlations were likewise consistent with previous work, both in terms of average and in terms of distribution. So turning to the first main analysis, we regressed choice probability against normalized neural sensitivity. While standard regression methods typically assume all data points are independent, this is not the case for our data. because we used chronically implanted electrodes. So we used hierarchical Bayesian regression to correct for the correlated measurements. Essentially, this performs separate regressions per session and per electrode and stitches the data together. In the example shown here, we see a weak but significant correlation. But when we look at the rest of the data set across both monkeys and both task types, we see no significant relationship between choice probability and sensitivity consistently. So to summarize a few key points, for chronic array data, it's really important to account for the fact that measurements may be duplicated when stitching together data across days. We addressed this using hierarchical Bayesian or mixed effects regression, which is really important to avoid inflating significance. And second, we do see some interesting structure in here, but I've just been told my time is up, so I'm going to skip that part. We had a second main experimental test about whether or not we see F prime structure in the covariance matrices based on projecting the covariance in each task onto the two axes. Long story short, we also found no results there, but in the interest of time, I won't describe these plots. So to summarize, hierarchical inference predicts task-dependent F primes of sensitivity structure in both choice probability and noise correlations, which is supported by evidence from previous experimental work. But we failed to replicate these previous findings, which may or may not be a null result for the theory. More generally, this failed replication calls into focus differences with those previous studies in terms of differences in analysis methods, experiment design, idiosyncrasies of the monkeys, or even some unlucky sampling of the neurons. So thanks everyone for listening, and I'll be happy to take some questions. I'm going to leave up some FAQ and discussion points here. And if you give me one moment, I will get my entry for the costume contest, which is something that is both frightening and topical. So here you go.
What does it say? Okay, I'll just stop for a second your screen sharing just to take a screenshot because it's too fun. So Richard, thank you for the talk. Let's start with the questions. I have a question. So since one thing that I think you mentioned that it was different from previous studies is that you decided to use the whitening, and it was effective in reducing spurious, in reducing artifacts.
Yeah.
Have you tried to use something different and something less effective to see if actual previous results may actually be based on including also artifacts in the analysis?
Yeah, I mean that's where we are on this. Is we tried both. We tried not whitening and we tried whitening, and I can tell you on our data, the main difference is sorry, let me find this slide. So yeah, what we find is that this reduces the number of like millisecond level coincidence spikes that we see across channels, which would be totally unexpected given the array spacing. So there's some additional like overall fluctuation that's shared between them. And I can point to this paper by Schultz et al. In 2015, which found that the single biggest contributor to correlations in this type of array was shared local field potential fluctuations. But in that paper, they did not try any methods to reduce those correlations. And what we've done is whitening, which we find empirically gets rid of those coincidence spikes across channels.
Okay.
And we haven't done something more systematic than that.
Yeah, no, it's in any case, it's interesting that here, since your results look quite clean, it's interesting that then you get something very different from the literature. And of course, if the participants or the panelists want to jump on, please do. In the meantime, you mentioned in the abstract also histology, am I right? Did you skip it for?
Yeah, that was for time.
Okay. No, just because I'm curious on how you used it.
Yeah, so this is a brand new and unpublished experimental method that Rick Bourne is working on. Basically, they were able to cool down the cortex to the point that they could extract the probe and get an estimate through slicing of where the tip of the probe was. And what we found was that in monkey U, the majority of the neurons were in layer 4, where like the theoretical predictions, and I think what most people have in mind when they look at feedback in noise correlations and things, it's layer 2, 3. We don't yet have the histology data for the second monkey though, so we can't say too much more about that. Oh, and the other thing I can say is, even though the predictions are more likely to hold in layer 2, 3, if we just rerun everything on the layer 2, 3 neurons in monkey U, we still see null results.
That's quite puzzling. How did you, so I imagine that as you got these results at first, you would have thought I probably have done something wrong, right? Did you do any troubleshooting to see, to actually be convinced that there may be something bigger than just small issues?
Yeah, this has been a long process of convincing ourselves that there's nothing here, and part of the problem is this one data point. We have essentially a very small fraction of the data that looks exactly as expected, but everything else is just kind of not there, despite no other apparent differences between those sessions. And so we've applied lots of different analysis methods. We've tried direct replications of previous studies that I don't really have time to go into now. But you're absolutely right that it did take us a long time to realize that this ultimately is a null result.
So did the previous studies looked at a similar amount of monkeys and a similar amount of channels per monkeys, or?
Yeah, so on this discussion point, so should we even expect to see choice signals in V1? There are basically 2 previous studies, each with 2 monkeys, because that's the standard. So 2 studies that say, yes, there are choice signals for this type of stimulus in V1. Hendrika Nienborg has a separate paper where she uses a different type of stimulus in V1 and finds no choice signals. It might have to do with the stimulus, but we use the same stimulus as these 2 papers. But there was another recent paper by Jasper et al. In 2019, which also, I think, used a similar stimulus, don't quote me on that, but did not find choice signals in V1. So currently, if you account for our study, that seems to be a tie for 2 yeses and 2 nos on whether or not these should be there.
Okay, no, that's interesting to hear. And actually, Etienne, if you want, of course, you can just ask Richard.
Yeah, I have one small question for you. What do you use to build your hierarchical Bayesian model? You're using R to make it?
Yeah, well, we use Stan, which
okay.
Yeah, which everyone who uses R uses Stan, but we put Stan inside of a MATLAB pipeline. So ultimately, it was a Stan model for the hierarchical regression.
And do you think a Bayesian model, they can easily be used with time series data? I'm really not so familiar. I'm reading about it because I think I would like to switch in the future. But for the moment, it's not really clear to me how to use it, for example, with the time series data. Say, for example, that I want to find if there is an effect somewhere. Is it possible to use this kind of model?
What kind of time series data?
For example, electrophysiological data.
Yeah.
For example, because for the moment, what I can have with my p-value is I can say if it's significant, but I cannot say if there is an effect or if there is no effect like you will have with the Bayesian factor. So that's why I'm more and more interested into this and to draw more reliable statistical conclusions. That say, I'm not sure how it works with time series data.
Yeah, I mean, okay, let me say one more thing about this. This is kind of a non-answer, but maybe it will help a little. So there's a lot of data processing that goes from the raw data to the points that we end up regressing, right? So we had, so we have this regression here, but each one of these data points reflects a large number of trials for a single neuron, which involves slicing out the right trials based on signal strength and all this stuff. In principle, it's possible to build one big statistical model that goes all the way down sort of to the raw data. Instead, what we do is we pre-process the raw data to get these XY coordinates in this space, and then we apply the statistical model on top of that.
So it means that you might are losing some information at the single trial level to extract some statistic about your data and then build your model on top of it.
Yeah, so the way we address that is, I mean, we have an estimate of the individual uncertainty in the X and Y coordinates of each of these individual points, which we get just by bootstrapping the data. So by resampling the trials, we can estimate our uncertainty in each measurement, but in the end, we're applying the statistical model just to the two-dimensional regression instead of the high-dimensional like time series data. Because my experience is that Stan is great for this kind of thing, but it requires much more hand-holding, if you will, to apply it to a larger, richer, time series data.
Okay. Thank you.
Okay, so it's 46, so we